Hello, my name is Shane Dunn. This is my muscle contraction PowerPoint for my BIO-515 class. If you are my students for my other classes, this is for my anatomy classes, this might be helpful for you. This PowerPoint is mainly on the muscle contraction, so sliding filament theory and such. But before I go on, I want to do some background information. So this right here, I will use red, is what we'll, oh, no, sorry about that. There we go. So this right here is what we'll consider a muscle. Inside a muscle are a bunch of these weird looking blobs. These right here are going to be considered fascicles. So you can pull those out and you pull this fascicle, which is showing. And inside each of those fascicles are going to be a muscle fiber or a myofiber. Myo just means muscle. So you might see myo instead of muscle. Please know the difference and please know that it's the same word, and depending on your teacher and how their test is going to be. There we go. And then in the muscle fiber or myofiber, we have these myofilaments right here. And then we have these things right here. You might know what those are, but we'll see those in the next slide. So here is going to be a more close-up look. So skeletal muscles, I said, broken into skeletal muscles, then fascicles, and then myofibers and myofibrils. Fibril is the smallest. So skeletal muscle is striated, as you can see in this top picture right here. See how it goes dark, then it goes light, and then it goes dark, and it goes light, and it goes dark again. That is striation. Those striations are important. We'll talk about what those are in a little bit. We also have mitochondria. Muscles need to do work, so they need to be able to make ATP and energy. And those are where the mitochondria come into play. The next big thing I want you guys to understand is the triad. So this isn't the Chinese triad, the gang. Tri just means three. So these are three structures, and I'm going to use different colors. So let's use green first. So the first structure is going to be the thing I'm highlighting in green, and I'll kind of shade it in right here. That right there is called the terminal cisternae. That's called terminal because it's at the ends. You have one on one side and one on the other side. This is terminal cisternae. And then I'll use red. You have this next you have this yellow thing called the T-tubular tubes. Either T-tubes or T-tubulars or tubules, depending on who your teacher says. I see T-tubes, it's much easier to say. But here are T-tubes. And then we have, I'll use, well, kind of purple, with this big blue thing in the middle between both the T-tubes is called the SR, or sarcoplastic reticulum. And I'll do that in purple. It's this thing that I'm kind of shading in purple. So that's the triad, and I'll explain what the portion is, so we know what that looks like, as I mentioned that. So up next, we're talking about sarcomere anatomy. So this is going to be the individual filaments from the myofiber. So these are the myofilaments. So if you can see this right here, these little dots right there, we're individually pulling those out right there. Okay, so we have actin, which is the blue. We have myosin, which is the red. And as you can see, myosin is thick, and that's going to be the dark band. I told you again how it's light, dark, light, light, dark, the striation. That's because myosin is dark and actin is thin and light. You need to know the difference because they play an important role. A lot of test questions in class will be based off this. Now, the sarcomere is from this Z-looking disc right here. From Z disc to Z disc is one sarcomere. We have millions and millions upon millions of sarcomeres in muscles. And then we have different types of bands. So for example, the I band, which is between actin and actin, where there is no myosin, that's going to be our light band. Has I, that's why it's called the I band, it's light. The A band is where myosin is located, all the dark band. And dark has A, so myosin is the A band. The M line is just dead middle. The H zone is where there's no overlapping of such. So that's the sarcomere. We'll talk about that in a little bit. On top of the actin, these little balls right here are actin, we have two types of protein molecules we need to talk about before we continue onwards. The first one is trypomyosin, right? So we know trypomyosin, and the trypomyosin blocks the myosin binding sites. So that's why it's called trypomyosin. Know that it blocks the myosin binding sites. The easiest way to remember that. On top of this rope-like structure, which is the trypomyosin, I'll use red in a second to switch my color. There's a red right here. That is the trypomyosin, that rope-like there, and it's kind of blocking these little dots. Is we have troponin. It's a surface protein molecule receptor that sits on top of trypomyosin. And what happens is calcium comes, binds to troponin, causes a conformational shift in the protein structure, and that kind of moves it, allowing the myosin binding sites on actin, sorry about that, to bind. Okay. Now that we understand all that, now we're going to talk about how a muscle contraction starts. And we're going to start off by drawing the picture. We're going to draw the picture of the neuromuscular junction, or motor end plate. Now, we kind of talked about this with neurons before, but it's pretty much the same thing. So I'll draw the end of the axon to the terminal button. Okay. As the action potential comes down, I'm going to use that in yellow, so it's basically writing. As the where's my mouse? How about that? Okay, so as the action potential comes down, right, it's going to open up voltage-gated channels on the terminal buttons for calcium. So calcium is going to rush in. It should all be reviewed from the neuron. So calcium is going to rush in. When it rushes in, it's going to meet some vesicles or bubbles. These vesicles are slightly positive, so all the calcium rushes in and kind of pushes them out. And inside these vesicles, a bunch of neurotransmitters. In the case for motor end plates for muscles, the neurotransmitter is acetylcholine, or ACH. You don't want to write acetylcholine because it's too long. We're always abbreviating to ACH for acetylcholine. And then as the calcium rushes in, these guys get pushed out. This process is called the exocytosis. Exo means exit, cytosis means cell. So all the acetylcholine neurotransmitters leave the cell and they end up in the cleft between the motor neuron, that's a muscle, causes movements, motor neuron, and the muscle, which I'll draw right here. They're not touched, they don't touch, but they're very, very close. On top of the muscle we have receptors. And those receptors will accept, I'm just trying to draw them really big, will accept the acetylcholine. Now, these receptors are a very, very interesting name. They are Nicotinic acetylcholine receptors. So they respond to two things in their name. One is acetylcholine, and the other is nicotinic. So nicotine can also cause this. That's why nicotine can cause sometimes muscle stiffness if you smoke too much nicotine or so at a time. So when the acetylcholine binds to its receptors, it's going to cause a gate to open up, a receptor-mediated gate on the cell protein, and that's going to let in a bunch of sodium. This should sound familiar when neurons talk to each other. It's the same thing pretty much. So a bunch of sodium is going to come rushing through. And that's going to make the muscle cell kind of have an action potential. But the muscle cell does not have an axon, so it goes down the T-tubes. So it goes in the T-tubes. What's kind of special with T-tubes is they wrap around almost all of the fascicles, and they kind of branch out. And when they branch out, they kind of run into terminal cisternae and the SR. More importantly, we're going to worry about the SR. So the action potential is going to go down the T-tubes. As it goes down the T-tubes, it's going to kind of go out, go all over the place as lightning does, and it's going to touch the SR. And the SR kind of crosses all over the place, on the surface area, has a bunch of calcium in it. And when the voltage gate, when the action potential muscle goes to the SR, it's going to be all its calcium. That calcium is going to go into the actin, go to the troponin on the tryptomyosin, bond to the troponin, move tryptomyosin out of the way, and allow actin and myosin to touch each other. That's what it kind of looks like in a better place, but it's better to draw it like I did. Now, I kind of skipped ahead a little bit, right? So this is the theory of how muscles move. It's called the sliding filament theory. So it's a cycle, so you can start anywhere. I like to start off where myosin and actin are already touching. So right here, myosin is already touching actin, are already in contact, and it has ADP and P. Now, ATP is the energy we have, right? Well, ATP, when it breaks down, becomes ADP, right? Plus phosphate. That right there is basically just saying energy is boost. So that P is surrounded by energy. Well, that energy is going to cause something called the power stroke, which is going to cause the myosin head to kind of move the actin and kind of slide it over it. So what's going on when a muscle contracts, it actually is shortening. And you kind of see this when you flex your bicep. If you flex your bicep actually gets smaller. That's why it kind of groups up and becomes bigger. And what happens, it releases ADP and phosphate, the power strip, right? Well, now it's stuck, right? Because it takes energy to do something. So we need ATP to attach the myosin head to pop it off. And then we break the energy on the ATP to cause it to reattach, knock off ADP and phosphate, and cause the power strip to occur. And this keeps on happening as your muscles get shorter and shorter and shorter, contract. It's kind of weird, kind of thing to think about. We kind of think like a rower, right? They have to start with the paddles in the water, and then they move, and then it goes out of the water and goes back in the water, and they move. It's called the power stroke, or kind of like what a rower does. Now, put it all together. So we have this drawing, and I'm going to draw the next part. So I'm actually going to use zoom for this. It makes it easier to draw myosin actin. So I'm going to draw the actin. And then a smaller one, and we're going to draw, oops, that's too. We're going to draw lines. And these lines are going to be the tryptomyosin. They're blocking the myosin actin sites. And then we have little dots on tryptomyosin called troponin. And we want calcium to bond to the troponin that moves tryptomyosin out of the way. So now we should not have any of the green dots, the green lines anymore. That will allow myosin to bind to actin. Yeah. Now it's already attached. So when it's attached, it has ADP and a phosphate. It's going to use all this energy right here to form a power stroke. So these heads are going to move backwards. And then it's going to get caught in the power stroke position. And it's going to need ATP to come pop it off and then back and forward. And that right there is a muscle contraction in a nutshell, simplified. So, sorry about that. I'm going to remove those drawings. There you go. So, here are some questions that make you think. So, Biffy Bob is a farmer who inhaled pesticide. Oops, I did forget to mention one key part that is important in GCU's anatomy class, depending on who your teacher is. If you have Wireman or Pramack, he emphasizes this guy a lot. So we've got to draw Pac-Man. He's already used yellow, I'm going to use an orange color. But this is an enzyme right here. I draw him like Pac-Man because everyone knows what Pac-Man is. That's Pac-Man. And his name is ATP. No, not ATP. Sorry, acetylcholine. Oops, 1 second. Sorry, I am being called. 1 second. Sorry about that. I was being called by a family member. So he is called not ATP synthase. I'm bad. Acetylcholine esterase, or acetylcholine ester. Esterase. And what his job is to do is to eat acetylcholine. His job is to eat it and neutralize it. So he comes down and eats all the acetylcholine and he puts it back into the terminal knob. The reason this is important is if we don't have this Pac-Man, this enzyme right here, muscles will keep on contracting over and over and over again. That is called muscle rigidity, and that's going to be called tetanus. Complete tetanus is what it's referred to when muscles can't stop contracting. So Billy Bob is a farmer who inhaled pesticide. So what are the signs and symptoms of this? And a hint, pesticides are mainly an acetylcholine esterase inhibitor. So if Pac-Man is dead, so Pac-Man's dead, he can't do his job anymore, acetylcholine is going to constantly keep binding over and over and over again to these receptors. It's going to cause muscle contraction. It's going to cause a muscle contraction. It's going to cause a muscle contraction. There is no relaxation. That's complete tetanus. That's bad. Okay. Myasthenia gravis is an autoimmune disease that destroys nicotinic acetylcholine receptors. So what that disease does is it actually destroys these receptors. The receptors are not there anymore. If those receptors are not there anymore, acetylcholine can't bind. Acetylcholine can't bind. Sodium can't come in. If sodium can't come in, can you have a muscle contraction? Yes or no? Hopefully you said no. And that's going to be called flaccid paralysis, where you go limp and you can't have contractions. So Botox is a cosmetic procedure that causes muscles to relax. It inhibits the exocytosis of acetylcholine. Well, exocytosis is this part. So if this part stops, the muscle contraction even occur? No. That's why botulism stops people from showing wrinkles because their face can't contract. Why bad Botox makes people look kind of weird, like they can't express. There we go. And there's my references. Hopefully the video was helpful. Thank you very much and have a good day.